the screen already, we have Lori Tessler,
Hi.
research technician in Dr. Fernie Howe's lab from the Division of Neurodegenerative Disorders. Thanks for joining us today, Lori. How are you?
Doing good. I'm doing really good.
Awesome. So, we've interviewed a couple of technicians this month, but if you wouldn't mind going over what is a technician? What does a research technician do?
Oh, technicians are the helpers in the lab. We'll get a variety of students that are doing their master's or their PhD. We also have postdocs who have completed their PhD and are now working in the lab. And we also have research associates, lab manager. And what we do is the technicians help the students with their projects, the postdocs, and the regular upkeep for the lab is kind of what technicians do. We upkeep in the lab, seeing what buffers and solutions are running low. Someone will come and say, oh, this needs to be made or this has to be done. And then I step in and get it done.
Awesome. So you're sort of making sure everything is nice and stocked, but you're also on a bunch of different projects.
Yes,
What
yes.
kinds of projects are you doing right now?
The focus in our lab is diabetic neuropathy. So we focus on type 2 diabetes research. And I actually have my own project I am working on with my PI, but I also help everybody else that's in the lab. And the focus for the one that I do the majority of is in type 2 diabetes research. And I do primary cell culturing, Western blot analyses, fluorescent microscopy.
Could you tell our students what a Western blot is and what fluorescence microscopy is?
Okay, well, fluorescent microscopy, I'll start with that. It's a microscope where you're going to see images that fluoresce. So, anyone, I guess the best way I could think of when someone is thinking of fluorescence, they think of the black lights, they think of that glow in the dark
Yeah,
sort of
like that
effect.
neon color.
Yeah, glow in the dark neon colors. Yes. Yeah. And what we do is we take images of tissue or cells, we apply color to them, and how we do that is with an antibody that has a color attached. It's more, there's more to it than that, but when that attachment happens, then these microscopes can pick up that color and you get to see it on the screen.
So what you're saying is you have something in a cell, and then these little antibodies come on with a little sort of light fluorescent thing
It's
on there.
called a fluorophore attached to it.
And then they find exactly where they're supposed to meet. And then when
Yes.
they meet and you shine a light on them,
Yeah,
it glows.
that's right. Yes.
So then,
It'll have a certain emission point, and the microscope can pick up on that emission on different channels on the microscope.
so then you can see exactly where certain proteins in a tissue are.
That's right.
Perfect. And then we're learning a lot today. What is a Western blotting?
Oh, Western blotting, it's one of my favorite things to do in the lab. It's in the study of proteomics. It's protein, all about protein. And we do what's called SDS page electrophoresis in the lab. So we separate proteins based on their molecular weight. And we go a step further and go into the blotting process, where after running proteins on a gel that run based on their molecular weight, we transfer them to a blot. And that's where the term Western blotting comes from. There is, yeah.
So then what? So They're different sized proteins and they're sort of falling through the mesh, and the smaller proteins sort of settle to the bottom quicker than the big chunky proteins. So
Yes.
you sort of get a nice ladder of the different sizes of proteins throughout your Western blot.
Pretty much, yes. And it's exactly it. It's like a ladder. You can see all the proteins based on their size.
Awesome. Do you do any other cool things in the lab that our students might like to hear about?
Um, I'm starting to dabble in mitochondrial research. So we have two different machines in the lab uh, with cool names, actually. We have what's called the Seahorse XFS and XF analyzer, and we also have what's called an Ouroboros instrument, which is an oxygraph. And it's to look at the function of the mitochondria in the cells. And I'm starting to work on the Ouroboros here.
So, as all high schoolers know, the mitochondria is the powerhouse of the
House of the
cell.
cell, yes.
So, you're just looking to see if they're working top notch or if some of the things we do sort of weaken our mitochondria. Is that what you're doing?
Yes, I can be a little bit more specific than that. I know high schoolers will, or they should know what's happening in the mitochondria. It's where ATP synthesis will occur. It's the energy currency of our cells. And I know in school they learn a lot about glycolysis and then what's called the Krebs cycle. And then what goes on to the inner membrane of the mitochondria, it's the electron transport chain. It's part of oxidative phosphorylation. The focus is on the electron transport chain. So there's glycolysis, there's the Krebs cycle, there's the electron transport chain, ATP synthesis, and I focus on the electron transport chain
So,
in the
you
inner membrane.
focus on the last part of making
Yes,
energy in the mitochondria.
yes, yeah.
And you and I barely remember those things.
Yeah.
So, how long have you been working as a research technician?
Since 2009.
Have you been working here since 2009?
Yeah, I started at St. Boniface Research Center, Albrechtston. At the time, it wasn't Albrechtston Research Center yet, but yeah, I started in 2009 in another lab.
Oh, were you also studying brains and mitochondria and all those things in the previous lab?
No, it was actually cardiac research. And then I moved upstairs to D&D, which stands for Division of Neurodegenerative Disorders. And part of it right now is type 2 diabetes research. I also have been in another lab where the focus was on Alzheimer's disease and gestational diabetes.
Oh, so I definitely forgot to define what diabetic neuropathy was for those who may not know. But when you get type 2 diabetes, the high sugar in your blood and stuff like that, your neurons don't really like it and they start
Yes,
to die. So
yes.
then diabetic neuropathy is that the dying of your like eye neurons and your
It's many forms. It's the pins and needles that you'll get, particularly in your feet and fingers, which can lead eventually to the death of the tissue, necrosis of the tissue, where people actually will have to get amputation of their foot, their leg, their hands. There's also glaucoma, effect of the eyes, which is a part of neuropathy. There is absolutely no cure. There is no medication. There's nothing for diabetic neuropathy out there. Um, it's a big problem, and in our lab we're looking at different drugs that hopefully can go onto the market that can start helping people who suffer from diabetic neuropathy.
So these medications that or treatments that you're looking at, they won't reverse the neuropathy, but they can help maybe slow down the process. Is that
Yes, yes.
awesome? So you looked at brains, diabetes, hearts,
Mhm,
Alzheimer's,
mhm.
jack of all trades.
Yes.
Did you have any other research jobs before you ended up here in 2009?
I did, but in a completely different field. Completely different field. I actually worked at the Canadian Grain Commission in entomology. So I was looking at insects in grain. So completely different field of research. Uh, I also worked at Central Testing Laboratories, which again, it has nothing to do with human disease or any sort of research, animals, nothing like that. Coming to St. Boniface, it was a completely different job completely. Yes, it was a new world. Yeah.
No kidding. So I guess as a research technician, you can probably study almost anything you're interested in. You've gone from bugs to brains to hearts to diseases. And
Yes.
so you can pretty much do anything that you're super interested in.
Yes, it is. I love being a technician. Um, I love science. I also like being able to make and do things in the lab, but also having stable hours as well. I do have a family at home, so I really appreciate a typical work week, which is Monday to Friday during the day.
Mhm.
There can be technicians out there who will work evenings and weekends, but I've been very fortunate to have a job where I get to have my focus on science during the day, and then I get to focus on my family in the evenings. So it's a perfect balance.
Awesome. So you get to do really cool science things, just like we
Yeah,
talked about fluorescence microscopy, Western blotting with the
yeah.
proteins. I'm sure you've done a ton of other things. And then you're pretty much a 9 to 5, Monday through Friday. And
Yes.
then you get to have your evenings and your weekends, but still
Yeah,
get to do something you love.
that's right. Yeah.
Awesome. So, where did you go to school and what did you graduate with to become a
Okay.
research technician?
I started out at the University of Winnipeg. I did my honors biochemistry degree, and from there, for a lot of people, the natural progression is to go into a master's program. But I had an opportunity present itself. And so it's actually quite strange. Most people don't go from university to Red River College, but that's actually what I did. So once I finished at university, I actually took a 2 year program at Red River College called Chemical and Biosciences Technology, and I graduated with that. I found university to be a lot of theory, and not as much hands-on approach. So when labs would happen, that would be the only time I would get near instruments, but it was more of a hands-off, like don't touch, just press this button, do this.
Don't mess
I
up the machines.
Yes, don't mess up the machines. And I really wanted to play with the machines. And this 2 year program at Red River, I found out about it. And I found out that on like they allow you to use a huge variety of instruments, and it was all about hopping on those machines, playing with the machines, actually working with them, troubleshooting with them. And they actually gave you hands-on training in aseptic techniques, in dealing with cell culture, which was rare. I didn't see that in university. And when I got to do that, it was literally a class where there was no paperwork, there was no test. It was hands-on training on how to work in a biosafety cabinet and how to keep a sterile environment.
Yeah, because that's something most people don't know. And especially even if you go to university and you don't really apply it, when you're trying to keep cells alive outside of the body, you need to give them a warmth, food, and oxygen. And you know who else likes warmth and food and oxygen? Bacteria.
Area.
So
Yes.
you need to be very careful not to get those things in your culture or they might take over.
That's right. And I absolutely loved the hands-on training that I got. It was quite a unique experience to the point that actually one of their exams, most students, when they write an exam, they'll spend 2 to 3 hours um, writing, answering questions, writing, maybe doing an essay. The exam that I got to have in this culturing course was 3 people watching me as I worked in the hood. So, how I put my gloves on, how I put my lab coat on, how I entered the biosafety cabinet, left the biosafety cabinet, what I did. Every little thing I did, they were watching me and they were grading me on it. And yeah, there was no writing anything down. It was all a process. And it's like a driving exam.
That's
The
a good
first
example.
part of your driving exam, you'll answer questions, but
Make
the
sure you know where the stop
actual
sign is.
yes, but the actual test itself is getting into a car and driving, and someone is watching you while you drive. Well, at Red River College, in this particular program, I got to spend over a year where at the end for my exam, it was being watched by 3 people as I was working in the hood. And all they did was not say anything, they just watched. And you find out afterwards how well you did.
Must have been a little bit of a scary experience, but I guess if you know your stuff, it shouldn't really matter.
Yes, yeah, it is nerve wracking when anybody is watching you. The second you have eyes on you, it's yeah, it's a little off putting. But
Exactly.
all I can do is I recommend that program hands down. If there are students out there that want to go beyond book knowledge, because like you got to know that it's very important to have that knowledge. You have to have a foundation. Um, but when it comes to science, that foundation is knowledge. But then when it comes to working in a lab, it's the hands on techniques that are really important. And
Hmm.
it's not until you get on the job that you really learn, um, you start learning techniques and you go beyond the books and you start applying things in real life.
So, for the high schoolers that are interested, what was the name of your program again from Red River College?
It was chemical and biosciences technology, but it might have a new name today. I don't know. They do change the titles of their programs, so I don't know if that name is still around, but it was called Chemical and Biosciences Technology. And it ended up being a co op program. And in that co op program, I was put here at Elbrexton Research Center. And that's where I got my foot in the door here.
Awesome. So, for those of you that may not know what a co op is, it's sort of like a work term, like an internship sort of thing when you're in school where you work in a lab in the field that you're studying.
Yes. Yeah.
Oh, we have a viewer question. You've worked in so many labs. What are some of the qualities or characteristics of a lab that make them run really well? What tips would you give to make a lab run well?
You have to have people who are willing to collaborate and work together. Um, this isn't a field of solitary research. You are going to need help. You're going to have to be able to discuss things. And it's not just one person. The lab is a hub of many people coming together and working. Um, and that's where the magic happens. is where everyone's got their role and you all come together to make a difference. So it's never just one person. So to have a good lab, you have to have the ability to work with others and be able to collaborate and troubleshoot together.
So everyone in that lab is sort of working towards the same goal. And
Yes,
it is a way better and easier to get things accomplished with a big group of scientists than just one lonely person being like, oh man, I have so many jobs to do trying to figure out this problem.
exactly.
Awesome. So let's get into the nitty gritty about being a research technician. What is your least favorite part about being a tech?
Oh, hands down, getting rid of the garbage. Their garbage accumulates. So we have what's called biohazard waste. So anything that comes from a human or an animal, so blood, tissue, things like that, you can't just throw away in a regular garbage can. So we have different types of garbage. We have chemical garbage, we have biohazard garbage, and then regular garbage that people think about. Though the two types of garbage, chemical and biohazard, yeah, that's not exact. That's the part of the job that I can take it or I can leave it there. Like if I didn't have to change the garbage, that would be pretty nice. If it just
Definitely.
could take care of itself. Yeah.
And could you tell our high schoolers why you can't just throw biohazardous material into the regular trash can?
Oh, yeah. You can't have anything that was um, alive and then no longer being thrown into a regular garbage can where, we do have cleaning staff here that come after regular hours to change our garbage, and for them to show up and to see possible uh, blood or you know tubes with that you can tell is tissue. It's just dangerous. You don't know. Every lab is different, so there could be drugs applied to the tissue. There can be toxic substances that have been tested. It's just not something that you can just throw into a regular garbage. Every lab is going to treat their stuff differently, and you can't automatically assume that what's being thrown away is safe. It's got to be done through a specific avenue to be
Mm.
properly disposed of.
So I might have drugs, poisons, or even
Yes,
diseases that you can get from these tissues if you don't
that's
know
right.
what's
That's right. Yeah. And we have to be very careful. And then we have chemical disposal. Every lab has to dispose of old chemicals and chemicals that they no longer, not old, but they've used it and now they need to get rid of it. And that is a process as well to safely get rid of chemicals because, again, you can't send or put something down the sink or send into the regular garbage. Number one, you can harm someone with that. Number two, you'll harm the environment, and there are safe ways of disposing of things. So there's a little bit of paperwork involved,
Mm.
and there is a process that we have to deal with occupational health and safety to get rid of what we have in the lab. And technicians, that's part of our job, is to deal with the biohazard garbage, and it's to deal with chemical disposal.
Mhm. And then do you dispose of chemicals that are like maybe flammable differently than those that are corrosive, can like burn your skin, or those that are carcinogenic that can cause maybe cancers?
The two biggest ones in the lab I'm in is carcinogenic substances. Um, we use a lot of paraformaldehyde, so that's an example of one, and a lot of corrosives, so acids and bases, so like hydrochloric acid, that sort of stuff. You got to be careful when you're dealing with those substances, and you can't just get rid of them down the sink. It's got to be taken care of properly.
Definitely. Now let's end off our interview with some advice for our young scientists. What piece of advice would you give them?
Take advantage of the co-op programs. If you're ever given an opportunity to work in the lab, the best thing is to be a sponge and absorb as much as you can, because it's on the job. It's that training that is so valuable, and that goes beyond the textbooks. So my recommendation is, if you're with your higher education, to try to maybe get into a program that does offer a co-op,
Mhm.
because work experience is invaluable.
Definitely. A lot of people I've gone to school with, they've done the co op program and they got to work at super cool places like the National Microbiology Lab and hospitals, working with plants and agriculture, studying animals in the ocean. Like, you
Yes.
could do whatever you want.
Yes.
Great piece of advice, Lori. So, thank you so much for joining us for our weekly Meet a Scientist interview. I hope you had a great time.
Thank you. You have a good rest of the day. Thank you for the interview.
Thank
Bye,
you.
everyone.
Next week is spring break. Woot woot. So we won't be having a Meet a Scientist next Thursday. But make sure to tune in after the break for some more Meet a Scientist interviews from the Youth BioLab. Make sure to subscribe to our YouTube channel for updates. Check out our website at youthbiolab.ca. And make sure to follow us at Youth BioLab on Instagram and Twitter. Thanks for watching, everyone. Talk to you next week.
Okay. Bye-bye.
Okay.